So this is a continuation of chapter 13 of the peripheral nervous system. So here I'm discussing the regeneration of nerve fibers. So as you probably all know, that mature neurons are amitotic. In other words, they do not undergo mitosis. Once we have a neuron, that's it. If they die, it is not replaced. But if the soma, which is the cell body of the damaged nerve, is intact, so if you have a damaged nerve, the peripheral axon may regenerate in the peripheral nervous system. Unfortunately, this does not occur in the central nervous system. So, with regards to the central nervous system axons, most CNS fibers never regenerate. And the reason being is that the oligodendrocytes, which are the myelinated cells that wrap around these structures, they bear a growth inhibiting protein that prevents the CNS fiber from regeneration. So right there off the bat, it's a preventive measure. Astrocytes, at that injury site, they form scar tissue. So that's another hindrance to potential regeneration. Possible treatment is neutralizing the growth inhibitors that are generated by the oligodendrocytes, blocking receptors for inhibitory proteins, or destroying scar tissue components. So in the peripheral nervous system, the axons, they can regenerate if damage is not severe. So we have axon fragments and myelin sheaths distal to injury can regenerate, which is Wallerian degeneration. The degeneration spreads down the axon. Macrophages clean the dead axon debris. Schwann cells are stimulated to divide. Axon filaments grow through this regeneration tube, and the axon regenerates and new myelin sheath forms. So here we have the axon fragment. The cut axon ends seals themselves off. The axon transport is interrupted, causing the cut ends to swell. Once that, without access to the cell body, the axon and its myelin sheath begins to disintegrate distal to the injury. So here, this is where the impulse is coming from, and everything distal to this all the way to the end. So from here all the way, that portion of it will disintegrate. Degeneration of the distal end of the cut axon, it's called the Wallerian degeneration, and spreads down the axon. Schwann cells and macrophages clean out the dead axon distal to the injury, and the surviving Schwann cells engulf the myelin fragments and secrete chemicals that recruit macrophages, and these macrophages help dispose of the debris and release chemicals that stimulate Schwann cells to divide. Section: a lining of Schwann cells form regeneration tubes. So here, Schwann cells release the growth factors and express cell adhesion molecules, or CAMs, that encourage axon growth. So here you could see the fine axon sprouts or filaments that begin to bridge over where the insult occurred. Schwann cells line up along the tube of the remaining endoneurium, forming a regeneration tube that guides the regenerating axon sprouts across the gap to their original contacts. And then finally, the axon regenerates and a new myelin sheath forms. The Schwann cells protect and support the regenerating axon and ultimately produce a new myelin sheath. So now I want to talk to you about components of a reflex arc. So reflexes. So components of a reflex arc, which is actually the neural path. So we have the receptor, which is the site of the stimulus action, sensory neurons, which transmits afferent impulses from the periphery into the CNS, and then followed into the integration center, which is either monosynaptic or polysynaptic region, within the CNS, and then followed with a motor neuron which conducts efferent exiting impulses from the CNS, from the integration center to the effector organ, which would be skeletal muscle, and so forth. The effector is the muscle fiber or gland that responds to efferent impulses by contracting or secreting, respectively. So reflexes are classified functionally as somatic reflexes and autonomic visceral reflexes. The somatic reflexes activate skeletal muscle, and the autonomic visceral reflexes activate visceral effectors, essentially smooth or cardiac muscles or glands. So here this image shows you a nice representation of this reflex arcs. So here we have a stimulus, a pin, it pierces the skin. Here we have the receptor, and the impulses is going from the periphery into the dorsal root, where this is where the dorsal root ganglia is, and then it synapses with in the integration center with an interneuron, which stays within the CNS here. We integrate, and as a result of that, it will then stimulate an appropriate motor neuron to send impulses out through the ventral root ganglion, and this will then go to its respective effector organ for the appropriate response to get out of harm's way. Spinal reflexes occur without direct involvement of higher brain centers. So in other words, the spinal reflexes do not require the brain, the cranium, to be able to respond and cause a reflex activity. The brain is still advised of the spinal reflex activity and may have an effect on the reflex. So testing of somatic reflexes is very important clinically to assess the condition of the nervous system. So if we have an exaggerated or distorted or absent, or may indicate degeneration or pathology of a specific nervous system region. So whenever we have any of these, most commonly assessed reflex are the stretch, flexor, and superficial reflexes. The next is the stretch and tendon reflexes. To smoothly coordinate skeletal muscle, the nervous system must receive proprioceptor input regarding 1, the length of the muscle, which is information sent from the muscle spindles to the CNS, and the amount of tension in the muscles, which is information sent from the tendon organs. So, functional anatomy of the muscle spindles, here in this case composed of 3 to 10 modified skeletal muscle fibers called intrafusal muscle fibers that are enclosed in a connective tissue capsule. The central region of the infrafusal fiber lacks myofilaments and are non-contractile. The end regions contain actin and myosin filaments and can contract. Regular effector fibers of muscle referred to as extrafusal muscle fibers. So here's an image showing you the anatomy of the muscle spindle and tendon organs. So here an enlargement of this, showing you this tendon organ and so forth. You have this annulospiral endings, which is the primary sensory endings, and we have the efferent motor fibers to the extrafusal muscle fibers here. The extrafusal muscle fiber is here. Intrafusal muscle fibers are inside here within this. And of course we have the tendon organ found in the tendon. So stretch and tendon reflexes here. Functional anatomy. So muscle spindles are stretched and excited actually in 2 ways. One is the external stretch, where external force lengthens the entire muscle. The other one is internal stretch, where gamma motor neurons stimulate the spindle ends to contract, thereby stretching the spindle. Stretching results in increased rate of impulses to the spinal cord. Next is the stretch reflex. Here the brain sets the muscle's length via the stretch reflex. So an example is the knee-jerk reflex, as I mentioned, where we strike the patellar tendon, and this strike reflex that keeps the knee from buckling when you stand upright. Stretch reflexes maintain the muscle tone in large postural muscles and adjust it reflexively. So in other words, it causes the muscle contraction on side of spine in response to increased muscle length, stretch on the other side of the spine for maintaining posture. Or as I demonstrated, when you're standing and you lift one leg up and you're trying to balance, so this stretch reflex is in full swing in order for you to maintain your balance and posture and maintaining that position. So how does the stretch reflex works? Stretch activates muscle spindle. Sensory neurons synapse directly with alpha motor neurons in the spinal cord, and the alpha motor neurons cause the extrafusal muscles of the stretch muscle to contract. In other words, the muscle on the outside. At the same time, we have reciprocal inhibition also occurs. Afferent fibers synapse with interneurons that inhibit the alpha motor neurons of the antagonistic muscles. So an example, in the patellar reflex, a stretched muscle, namely the quadriceps, contracts, and the antagonists, the hamstrings, relax. It's important to note that all stretch reflexes are monosynaptic. There's just one synapse, and ipsilateral, where the motor activity is on the same side of the body. Positive reflex reactions provide two pieces of information. One, it proves that sensory and motor connections between the muscle and the spinal cord are intact. And second, the strength of response indicates the degree of spinal cord activity. So pathological findings. Stretch reflexes can be hypoactive or absent if the peripheral nerve damage or ventral horn injury has occurred. Reflexes are absent in people with chronic diabetes mellitus or neurosyphilis and during coma. Stretch reflexes can be hyperactive if the lesions of the corticospinal tract reduce inhibitory effects of the brain on the spinal cord. So here, the patellar knee-jerk reflex, an example of a stretch reflex. So here, muscle spindle is stretched, and the impulse goes from the periphery into the CNS, integration here, monosynaptic, and then here we have one stimulus that's in red that will cause an excitatory to create and cause the quadricep muscle in this case to contract. At the same token, a simultaneous impulse is sent to the antagonist muscles, namely the hamstrings, the flexors, to cause them to relax, allowing the leg to extend out. And this is both excitatory and inhibitory responses. So just before was a muscle stretch reflex. Now this is the tendon reflex, and this involves polysynaptic reflexes, and what it does is it helps prevent damage due to excessive stretching. And this is important for a smooth onset and termination of muscle contraction. So here's the image here. So in this case, it is the quadriceps strongly contract, the tendon organs are activated, and the tendon is stretched. So with regards to this, impulse goes into the dorsal root ganglion here, synapse here, the afferent fibers synapse with interneurons in the spinal cord. Then The